Hello, good morning, good evening, everybody. I am Vaidya Asavari Manvikar. I would like to welcome you all to our GCAR Global Webinar Research Series. GCAR, Global Council for Ayurveda Research, is an international non-profit organization based out of USA. Our mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence based health science globally. You can learn more about us at www.ayurvedaresearchusa.org. Please do consider joining us as a member, which will bring you access to all our resources. No nonprofit can operate and succeed without volunteers. So consider joining us as a volunteer, and we can place you in a variety of projects based on your skills as well as your interest. Once again, our website is www.ayurvedaresearchusa.org. Now, let me introduce our eminent speaker today, Dr. Mitali Mukerji. She is PhD, FASC, FNASC. She is a professor in bioscience and bioengineering at IIT Jodhpur, affiliate faculty, School of Artificial Intelligence and Data Science, AIDE, and Center for Emerging Technologies for Sustainable Development, CETSD, IIT Jodhpur. She is a pioneer of Ayurgenomics, a very exciting field. Dr. Mukerji is a globally recognized scientist who pioneered Ayurgenomics, an innovative discipline that integrates Ayurveda with modern genomics to deliver personalized and integrative medicine. At IIT Jodhpur, she leads transformative initiatives including Thar Designs, studying human adaptation in the Thar Desert using multi-omics, Ayurtech Center for Excellence, building AI-driven, evidence-based integrative health solutions. She has 100 plus international peer-reviewed publications. She has been working very hard for many, many years in this field. Previously, she was also a chief scientist at CSIR Institute of Genomics and Integrative Biology. She led the Genomics and Molecular Medicine Department at CSIR's Ayurgenomics Initiative, Sutra. She also convened the Indian Genome Variation Consortium, which mapped the first comprehensive genetic landscape of Indian populations. Her work spans human genomics and rare diseases, biomarker discovery and disease susceptibility, environmental adaptations and mutational histories, AI enabled integrative precision health, which is extremely important in today's world. I'm very, very honored to have Dr. Mitali Mukerji here, and we are hosting this 6 series webinar series. And I would love Dr. Mitali if you can take it from here.
Sure. Thank you. Good evening, good morning, whichever part of the world people are hearing from. And thank you for giving me the opportunity to reach out to a large number of people about the research which has been carried out for the last 2.5 decades now, nearly. And I initiated this work in the CSR Institute of Genomics and Integrative Biology under the mentorship of Professor Brahmachari and Dr. Bhavna Prashar, who is an Ayurveda doctor, an MD. She and I developed this area of the last 20 years. So I would be giving in the 6 lecture series a kind of a historical way how we grew and how the area now is known in the country as Ayurgenomics. So something which we started in 2002 and kind of got the first leads, which gave the name to this area of Ayurgenomics, and now it's being widely used across the country and the world also. So I'll just start my slides. Just so is my slide visible?
Yes, it is.
Okay. I will. Uh, okay. So this is our campus where I'm residing at present. It's IIT Jodhpur. Anybody is visiting Jodhpur to visit us, and we now have a Ayush supported center of excellence in Ayurtech. And what was the need for having an Ayurtech will be clearer in the next series of lecture. I will start from the from where we initiated the work. So build it up from Ayurgenomics to Ayurtech. So so uh, if we look at a global health and disease scenario, and this is a study which was undertaken on data from 1950 to 2017 by WHO, it clearly shows that for all the meaning people across the world, what has happened very importantly is that longevity has increased. But if we look at the age in which diseases set in, a large number of diseases of non-communicable nature uh, is there across the last 10 years of a life. So even if the longevity has increased, the health longevity has reduced by more than 10 years. And it is with respect to many, many disease conditions. And the number is a staggering 46.6%, which actually kind of exposes how much health burden we are living with, and the need of the hour is how we can actually increase the health longevity so that people are living for a much longer time, and what are the ways we can provide solutions to enable this to happen. So now I would like to emphasize where the problems are from a perspective of my personal health journey, but it's a journey which many people would connect to in one way or the other. It's just a way to show how, as in the population, we as individuals take health trajectories, and why it might be important for us to understand our health trajectory if we want to intervene in a personalized manner. So, the early years of my life I spent in small townships, so not very stressful as in the metropolitan country, but there were frequent displacement because of the nature of my father's job. And then I suffered from some conditions which many people of that age group would suffer. For example, severe sweating, acne, braces. And for all these, I went to different doctors, or sometimes we had home remedies, whatever it is. For example, a thing like severe sweating, you don't go to any modern doctor. Braces, you can get your teeth fixed. Acne, people on the street or dermatologist would treat you depending on what works for you. And these are all different conditions which are treated by people. And then the later part of my career, when I began, it happened in Delhi, which was urban, and setting up institute with genomics, meaning I was one of the first person to join in this initiative of establishing the first genomics institute in the country under mentorship of Professor Brahmachari. So it was a career full of ups and downs. And then I suffered a condition called AVM, bleed in the brain, which was treated by uh, a neurosurgeon. and then there was onset of obesity, diabetes, fibroids. And during that time, I happened to meet an Ayurveda doctor, Bhavna, and other colleagues. And that time I was treated by for 15 days by a medicine from Ayurveda, and the sweating problem completely disappeared. So I kind of was living with something for 30 years of my life, and with the Ayurveda medicine that was maybe cured forever. And then there were other problems which were dealt partly by modern doctors, partly by Ayurveda doctors, depending on the nature of this. And now in my late 50s, I moved to a desert area, and I'm aging, and then this is a dry and a hot area where I come from, maybe the wetter part of the country, and the diabetes is managed still, and then there are newer problems which are cropping up. So if I were to look at all these kinds of things, everything was treated in a different manner. But if I ask an Ayurveda doctor to map this, maybe they might have a particular way to understand the history, and say that if I have to live further in a with a longevity, with a health longevity, maybe it's something of the inflammation which needs to be treated, or maybe the aging component need to be treated, but there is a personalization need. Now, this is where precision medicine comes in. That is, this is the modern buzzword also, is that how you can have predict a person's in a holistic way, from the time the person the inception has happened to prenatal stage, right till the till increasing the health span. So how you can plan a baby, if there is an inborn error, how you can rectify it, how you can prevent a disease which might run in the family, or which the person is at risk, how you can manage infectious disease, how you can target cancer, how you can customize so that you are able to maintain a good quality of life with multiple medicine, how you can increase the health span. And then if you know the cause of death, maybe you would be able to maybe delay or maybe understand why people die. So from pre-womb to tomb is where people are now thinking that if at all levels we knew our makeup, we would be able to have a much precise way of managing. Now, where does this thought crop from is from the foundational knowledge of our common organizing principle, which is in the DNA and all life forms on Earth. So, this was propounded by Francis and Crick, but nearly all forms of life have the same language. We share. If you look at bacteria, look at humans, look at mouse, rat, plants, all of them, the whole life is encrypted in this four-letter code ATGC, and their order or the way they are organized would determine the kind of species or the kind of biological intelligence which each of them show. And the fact that they share the same we all share the same unifying language is what allows a bacteria to infect us, a virus to infect us, COVID to grow within us, microbes to live in harmony with us. Because if we are not seamlessly having the same language, we cannot cross talk, then either of them cannot survive, right? So this is the universality which is there in all life forms, and this is where the genome comes in. So these are some of the facts that we, even though we have 10 to the power 14 cells in the body with different organs and tissues, all of them have the same genetic material residing in 23 chromosomes. And if we open it up, it will be like a book of ATGC written a billion times. And basically, this can be shown as an analogy. Just like you have the alphabets, you have these four chemical letters, which, when threaded, becomes DNA, which is like the words. and if these have punctuations, then they form genes, which is like sentence, and then a group of sentences will make up a chapter, which is one chromosome. And then when entire sets of chromosomes are threaded, it becomes a book. So this is the book of life of 3 to the power 9 base pairs, which we inherit from one parent, and one such book we inherit from each of our parents. So one book is called the genome, and the science of understanding the meaning out of these letters is the science of genomics. So the same book of life is shared across, let us say, the all species, because of which it describes that this is human, this is rhesus monkey, this is rat, but we are different, right? We though we all look human, we have variability in us, and this variability is because of these variations in the genome, which is because of when the DNA replicates, then it can incorporate certain kinds of changes, which if doesn't get rectified, then it is passed on to the next generation. And these could be one letter change, multiple letter change, a block of letter getting changed, something getting inverted, something getting duplicated. So, for example, if I draw the analogy from a word, if you have color, then you can have a deletion of a letter. It will have the meaning, but it is definitely a different spelling. And then there could be an insertion where there would be a word which has no meaning, and then it has to change to have a meaning. So it could be substitution, but then it will differ from what it originally started. Then there could be deletion, then there could be another substitution which could still have the meaning, but in a different kind of a lingo. Now, like cool is written in a different manner. There could be an inversion, and then there could be fusion. So all these kinds of events happen. So what you start with and what you end, like a color to cool look, is how we have all evolved from, right from when we moved out of Africa to different parts of the world, different places. The genome changed depending on what kind of need was there, or just random changes as arising and then getting fixed. And that's how all the people in the world, uh, we have very diversity, etc. Now, what happens if you have this change? So as I said, if there is a sentence which is a gene, and if there's one change, so typically what happens is the DNA is transcribed to something called a messenger RNA, and then this messenger RNA is read it moves out from the nucleus to the cytoplasm, and three chemical letters are read. These are called as codons, and then the process starts from mRNA to the functioning unit, which is the protein. So basically, three letters are recognized, and an amino acid is incorporated during the process of translation. So all the subsequent three letters, depending on what codon is there, any of the 20 amino acid will start getting stitched together, and this is what will make a protein. So you can realize if there is a change in the DNA, then there would be an altered RNA, which would then maybe change the codon, because of which a different amino acid will get incorporated. And depending on how that amino acid behaves in the protein, it can even give rise to something as gross as this sickle cell phenotype. So you have this RBC, which is circular in nature, and there's one change in the beta globin gene from an amino acid glutamate to valine, and it actually gives rise to this sickle cell. So, like this, there are 6,000 to 7,000 rare diseases, uh, which are which are uh, which in affect a large number of people, and one nearly 1 in 5 person in the world is living with one or the other rare diseases. So the combined prevalence of rare diseases are like a common disorder. So in the quest to understand the entire book of life, so that we are able to have a reference book from which we can compare all the other genomes was what led to this ambitious project called the Human Genome Project. It was started in the late 80s. It took 13 years. And in 2001, we got the first draft of the human genome. It was an expensive exercise, but it was a very ambitious exercise. The challenge was to read each and every base in the human genome so that we all have a reference genome from which we do all the comparison. So this was called the Human Genome Project. And the interesting part about this is that once these technology developed, because of this universality of the genome, or of DNA in across all life forms, one could use the same technologies to sequence pretty much anything in earth, any kind of tissues. So typically what is done is a sample is isolated from any part of the body, like saliva, hair, semen, any skin, and then you isolate the DNA. It is there in all the tissues, and then you can quantify it. And then typically you do something called PCR, polymerase chain reaction, so that you can amplify it. And after that, you have these machines where you can, through some chemistry, read all these nucleotide one base pair at a time. So from the time it started, so there were uh, different generations of sequences. From 1977, we had our first generation. After that, the human genome sequence happened in such kind of a machine. And then over a period of time, there has been a massive evolution of the technologies as well as reduction in the cost. And now the time has come where we have handheld devices in which we can actually sequence one genome every day. So from what took us 13 years, we literally can sequence a huge number of genomes in very high throughput manner. So this has led to a massive amount of sequence generations, not only of different life forms on the earth, but also of different humans, in a quest to understand not only populations, but how the populations are related and how disease happens, etc., etc. So during the time when these studies were going on, we embarked also on understanding Indian genomes, because Indians are known for the diversity we harbor. So typically, this map is a linguistic map where large number of people speak Indo-European language, a fraction people speak Dravidian. Then there are populations lining the Himalayan belt speak Tibeto-Burman language, and then in the middle part of the country, there's another language called Austroasiatic. So if one were to ask, how do you know, or how do you relate the people of the country, so that how many reference genomes need to be collected in India to be able to know uh, 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 whether we have sampled enough so that the genomes are adequate to address questions of across the country. We first needed to develop a genetic map of India. Basically, the question is, do people who speak the same language, are they more related? Or are, if they reside in the same geographical area, are they more related? What is it which will tell us how people in our countries related? So this was called as the Indian Genome Variation Consortium project, and I was the convener of the project. And it was a group of 200 people who worked together to develop the first comprehensive genetic map of India. And we collected samples across the country, 55 populations from different genetic background, different geographical regions, different ethnicity, to see, ask a question, how people are related. And we came up after something which is called as population genetic study. So basically, we have these sequences of DNA, and we look at uh, the distance between two genomes, meaning how much similarity they share with respect to different places in the genome. And based on the more similar they are, they would cluster into groups. So based on all those studies, we found that Indian populations could be clustered into five major groups, where language was not something which related all of them. So it was more of ethnicity, geography, and language. So, for example, if you see in this place, it's a belt of Indo-European speakers, but you have different colors. Each pie chart is the place from where we collected 50 samples. And then if the pie charts are the same color, that means they are more related to each other. And if they are different, then they belong to different genetic cluster. So you can see populations lining the Himalayan belt are more related to each other. But if you look at the linguistic lineage, these are called as TB, IE, AA. That means they speak Tibeto-Burman, they speak Indo-European, they speak Austroasiatic language, but they are all more related to each other. And then if you look at this belt, they are all Indo-European, large population, they are more related to each other. In this area, there are Dravidian population and then there are Austroasiatic population, but they are all isolated tribal populations. They are more related to each other. And the Dravidian caste populations are little different than the Dravidian tribal population. But here there are populations which are like the Indo-European, and which was very interesting was populations in the far upper region in Kashmir are also more related to Dravidian population. And this matches with the historical account that as people, there were more invasions from the western frontier. Maybe the original inhabitants were either pushed down or pushed up, and they still at the genetic level are more similar to each other than what we think. But now, why does this matter? This why what does this tell? So this is something which came out of the study. So for example, if you look at this map, this is malaria prevalence map, and this is a particular gene whose variations differ in frequency across the country. And you can see this belt, which is malaria endemic, has one version of the gene, and the remaining part has a different version. This tells that the people in this part of the area, which is malaria endemic, since they are residing, they would have had to have protection from malaria. So maybe they have variations which gives them a kind of a benefit. And this can be taken up to see whether these beneficials are lesser in frequency in people who suffer from malaria. So that's what was followed up by a colleague. Or for example, this is a gene which is differentiates the populations in the high altitude compared to lower altitude. And this is a gene which is linked to high altitude adaptation, which I will describe in my next lecture. But this is something we get from such kind of a map. So it tells us that if you overlay the genetic map with other kinds of maps, then you might be able to know how people are related to each other. So this genetic map is the first comprehensive landscape of Indian population. This is also kind of displayed in the Indian Museum in Kolkata. So, but these are all population level similarity. Within each population, also people will differ. Within a family, people differ. So that's where the problem starts. That the Human Genome Project was embarked on to have a reference genome from where we could compare. But what came out was a major surprise that even between two related individuals, we would differ in 1 million places. So that will actually lead to so many combinatorial possibilities that from looking at the genome of a person, you cannot actually understand uh, the phenotype of the person. And the other part is that in the modern context, we do not understand health or understand individuality. Health is always contextualized in the context of a disease, that if you're not diabetic, you're not asthmatic, then you are healthy. And this still remains a challenge, even though the Human Genome Project was embarked in 2001. Now it's 2026. Still, we are grouping with how to describe human individuality. And why is that important? People have now been able to show, and these are studies out of Cochrane trials. This is in a site called Number Needed to Treat. Typically, supposing there is a drug for schizophrenia, you treat 5 people and only one person has an effective outcome. There's a disease for heart, meaning medicine for heartburn, and you treat so many people and only one person has a effective outcome. So this is called as number needed to treat. Here, NNT is 5, that means you treat 5 people and you have one effective outcome. Here NNT is 4, here NNT is even higher. So the higher the NNT, that means lesser number of people are effectively uh, kind of benefiting from that medicine. So these medicines come out of randomized control trial, but then they are used in large bulk of population, the inter-individual variability becomes very apparent. And these are typically done for the doctors to make a decision how likely the person is going to have a good outcome. And there's something called NNH, which is number needed to harm. So you need to reduce the number of number needed to treat, so that you're more effectively treating a person, and you need to have a large number of NNH, so that many large number of people, if they are treated, very few get the side effects of the disease. The other thing which also was surprising after the Human Genome Project was that typically diseases in the modern medicine are studied from an organ perspective. And when the genomes were sequenced of different diseases, there seems to be not a simple one to one connection of genes to diseases. Excuse me. But there were one to many corrections. So that means one gene was linked to multiple diseases, and multiple genes could be affecting a disease. So as a result of which, it is not an isolated organ and gene, but it's all a connected network. So that's where the modern medicine is now undergoing a paradigm shift to a systems level thinking, where it is now understood that if we have to treat a disease effectively, we have to have a more system biology approach. The other thing which is also now people are appreciating is not only the genetic part, but if you have an environment or any trigger, then each individual also displays a difference with respect to the response to the trigger. So an individual can be at a particular baseline and can progress to any trajectory depending on how he or she harbors the baseline genetic, or the population genetic, or the environment, etc. And this complicates matters. For example, even a simple disease like sickle cell mutation, where only one gene is implicated, the problem is not today in diagnosing, but the problem is that a sickle cell patient, depending on what other genetic background the mutation is, or what environment the person is exposed to, can land up to get a stroke or pain or hemolytic anemia, aplastic anemia, bone infarction. And it is very difficult for in the modern times to map who is going to land up in what. So it's not only the genetic, it's also the environment and also the host modifier, which can actually change the course of the disease. And the idea is how you can break this to be able to have a more effective outcome or more effective management of disease. For example, sickle cell mutation gets exacerbated in dehydrating condition, in inflammation, or in hypoxia, and that leads to all these kinds of precipitation. So now the world is shifting towards something which is called as P4 medicine: predictive, preventive, personalized, and participatory. And also the focus is shifting to how we understand wellness. And there's a very wonderful book by Leroy Hood, which talks about age of scientific wellness. The idea is that supposing we are able to capture an individual in all possible dimensions of omics and physical activity and all the other things which we are capturing by these instruments, and can we analyze the person and create a virtual twin of the person, and then if we understand the digital twin by simulation, then could we have a better understanding of how a person is likely to behave in a spatio-temporal dimension. This is a very expensive exercise. This is an unaffordable exercise for a population like India, but western part of the world, that is US, have embarked on these kinds of studies. It requires huge data, huge cost in terms of measuring multi-omic data, and also uh, big data capabilities. Now, uh, now I'd like to shift gears that all that I'm talking about of understanding health, understanding individuality, understanding a person, understanding trajectory is actually known in the Indian system of medicine, Ayurveda, 3,500 or 5,000 years back. And what is very interesting is that the aim of Ayurveda actually talks about maintenance of health in healthy and alleviation of disorder in diseased. Already, it is implicit that they are talking about individuality when they say healthy and when they say diseased. And these Sanskrit shloka, I think if the audience is Ayurveda, they would uh, already be aware of these. And so this is not new to India. Even the western part of the world has started acknowledging that Ayurveda knew about precision medicine and treatment were tailored to each person's prakriti or constitution. Now, uh, is it still contemporary? People ask if 3,500 years back the conditions were different, then how will the medicine be relevant today, right? So this was one study which was embarked by Professor Brahmachari in collaboration with an Ayurveda telemedicine center, Jiva Ayurveda. And what they have is they have telemedicine institute portal across the breadth of the country. So they had digital data on 353,000 patients by 300 Ayurveda doctors by in-person consultation. So Dr. Brahmachari decided to have a look at how for what diseases Ayurveda is approached and in which and they looked at it by age category. So this is a alluvial plot which displays whatever is most is at the bottom and whatever is lesser in prevalence becomes lighter and it is at the top. So as you can see, this is age group. So the skin diseases, people approach Ayurveda doctors in the 10 to 20 age group. Digestive is all through. Endocrine problems in the early age, and then in the later age, skeletal problems starts to happen in later age. So like this respiratory problem more at the younger age and then at a later age. So and 82% patient had disease chronicity for more than a year, and Ayurveda treatment offered complete or partial relief in more than 76% cases. So this was, I think, the first big data analysis to show in a very kind of uh, 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 big data by big data analysis, the data itself was revealing that a large number of people are approaching Ayurveda for multiple kinds of disease in different age group. And the global market of Ayurveda also suggests that it's actually contemporary. A large number of people are availing Ayurveda medicine. In also during COVID time, when people thought that it's a very emerging disease, for Ayurveda, it was business as usual because for every new disease which is described, they have to go back to their ancient text and make meaning out of the new disease. So, and they treat. So it's like business as usual. So the pandemic is described as Padudhvans, and even the descriptions are very kind of vivid, which talks about even though there is dissimilarity in physical constitution of human beings, still there are factors which are common to individuals whose vitiation leads to simultaneous manifestation of disease, having same set of symptoms leading to countries' destruction. And these are air, water, location, and season. This is like very vivid. And then they would evolve a medicine by something which is yukti or reasoning, so that you look at the disease, you look at the phenotype, you look at the feature, and then you are able to use a reasoning approach to be able to evolve the medicine. And pandemic, when which exposed the limits of modern medicine, a large fraction of people. So I did some survey across thousands of people, and I find 42% of the people turn to Ayurveda either by for prevention or even for cure, and there was 50 to 90% growth in Ayurveda market. So which tells that it's ancient, but it's still contemporary. And Why that is so? So I'll talk about Prakriti. There's more to Ayurveda, but start from the foundational principles, because I got into this area because of this interesting understanding of constitution types for understanding individuality, which basically is like looking at a composite constitution type of a person through anatomical, physiological, physical, and psychological attributes. All the populations in the world can be broadly binned into seven broad categories, which describe, or which kind of informs about the state of health, the likely response, the likely disease trajectory, and the way to individualize management of disease. And once the prakriti of a person is formed, it remains invariant throughout. So it's like invariant. You age, but your prakriti doesn't change. Then there is something which is very, very akin, like genetic makeup. And uh, uh, why that is so is basically from a organizing principle in Ayurveda, which is Tridosha. So uh, uh, Tridosha is basically it's it determines the baseline proportions of three attributes, physiological attributes, which are called as Vat, Pitta, and Kapha. Vat governs the kinetic component of the system, Pitta governs the metabolic component, Kapha governs the structural component, and depending on what kind of uh, proportions you have, the Prakriti is defined. And the interesting part is the disease is defined as a variation from the homeostatic state, which would be different for different prakriti type. So what is normal for pith would be disease for vat, and for kapha. So the idea is, if you understand the prakriti, then you would know from the dosha itself what is deviated, and that would allow you to appropriately choose a medicine to be able to bring down the dosha to your homeostatic level, and not everybody to the same homeostatic level. And this forms the foundation to the network medicine concept, that is Trisutra, so that the causes, the features, and the therapy are all described by doshas. And that allows you to understand, if a person has particular feature, what is the likely dosha in them. If the person has a particular disease, what are the, which dosha it is impacting more, and then accordingly also one can choose appropriate diet or appropriate therapy to bring down the dosha. So this, and then the functional attributes of doshas are also given, that Vata governs shape, cell division, proliferation, signaling, movement, cognition, networking, all the things which are movement, kinetic, transformational in nature. Not transformational, signaling and communication, all those things. Pith governs things which involve digestion, metabolism, energy, pigmentation, vision. And Kapha governs growth, structure, storage, stability. So when I came across these terms, it sounded like a standard biochemistry textbook, because we also describe any gene or any pathway in terms of these very attributes. Only thing is the language is different. And what was very interesting to me was that Prakriti is described as not only a consequence of what you inherit from your parents, but it is also consequential of prenatal exposures, a mother's diet, lifestyle, season of conception, which would govern a Prakriti of a person. And it's influenced by the ethnic background, the heritability aspect, the geographical aspect, the season, the age, and then the individual variability, which was really amazing. Like uh, modern treat everything as a different subject, but here only in one shloka in Charak, you have all these attributes uh, uh, governing individuality described vividly. And all my 20 years, I've just spent around this shloka itself, because once the person is born with a particular prakriti, then you know that what time of the day is most appropriate based on the inherent fluctuation of or dynamics of dosha across time, across season, across uh, uh, age. And that's how people can predict who is likely to have what problems at what time of the day or what season or what age. So this is the entire kind of pre-womb to tomb kind of a knowledge framework which is embedded in the Prakriti definition. The other important aspect is that even if we say Vat, Pitta, and Kapha sound like abstract, the it also mentions what tissues they are more prevalent in and what parts of the organ or system they would more govern, as a result of which it would converge into phenotypes through which one can understand what is the prevalence or of the person with respect to Vat, Pitta, and Kapha. So, for example, Vat governs the brain, the lower gut, bone tissue, ear, and that would convert to the way the person walks, talks, remembers, bowel, bladder, frame, memory, etc. Pith governs the skin, liver, blood, eye. So this would transform into these kinds of phenotypes, which can be measured, and then the cuff governs these tissues, and which would also be measurable here. So you have from the abstract Vat, Pitta, and Kapha a way to look at these, and these are the ones which then go on to defining a person's attribute or composite phenotype. So Vat, Pitta, and Kapha will be there in everybody, but if a person is more of Vat, then maybe the differentiating features will be more evident from examination of certain attributes, and that is how one can then assess a prakriti of a person in a composite way. So modern people have also got definitions of describing healthy people on the basis of anthropometric attribute, on the basis of how their skin burns or tans, on the basis of their morningness, eveningness, on the basis of physiology, metabolism, physical activity. But if we look at Prakriti, Prakriti is composite of all of this. And this is what is the important aspect, that from a foundational dosha, you have a connect to the gross phenotype, and that's how you describe the seven prakriti types, in which the Vat, Pitta, and Kapha people who have extremes of either one of them would have more extreme and distinguishing characteristic in terms of body frame, in terms of physiology, in terms of memory, in terms of other kinds of behavioral attributes. And diseases are also described by the doshas getting perturbed. So Nanatmaj Vikar, if single dosha, and Samanyaj Vikar, if multiple doshas are involved. And 80 such attributes are described for Vat, and 40 conditions are described for Pith, and then 20 conditions related to Kapha are also described. And in doshas are also kind of understood for pathogenesis of disease, which is called as samprapti. So the idea is, in a particular environment, if you have an accumulation of dosha, which where it is accumulated, it will actually manifest particular phenotype depending on its function and the host where it resides, which will ultimately lead to the pathogenesis. So understanding dosha and its phenotypic attributes become very key to understanding samprapti or mechanism of pathogenesis. And just like we are talking about prevention and all from pre-womb to tomb, for every disease, the Ayurveda describes it as moving targets, and you have 6 stages of intervention possibilities, clearly definable by phenotypic manifestation. So there are chances for early actionable intervention before the disease manifests into something which is very difficult to treat. So the idea is that since a framework was already existing, which is a very advanced framework, which is what modern medicine are looking for, the idea is that would you, if we are able to stratify healthy people on the basis of Prakriti, would we be able to then bin them into groups so that we are reducing the NNT and increasing the NNH, so that we don't treat these many people, we only treat the people in which it is most likely to be effective, and as a result of which can we arrive at a scale. Can you just excuse me for 1 minute? Hello, somebody has come. Uh,
Sure.
yeah, 1 minute. Yeah.
Sure, sure.
So, so this was the gap of our knowledge, and the idea was, can we bridge the gap by this? So this is the problem, that one is written in Sanskrit and one is written in modern language. How do you evolve a common language for a crosstalk? This is how we started. So the idea was that, supposing people are belonging to Vat, Pitta, and Kapha, and if we take individuals who are at the extreme end, would they have expression of genes which are more related to the different functional attributes which are given to each of them? Or if we study the genetic variation, would they have more predisposition in genes which are linked to hemorrhagic disorder or neurological disorder or obesity, etc.? This is a very ancient slide, but this is what we initiated in 2002. And the idea was that we develop a working hypothesis that if Tridosha Trishutra principles are right, then could we develop a working hypothesis based on the principles and test them in Prakriti or in cell model and try to see whether there are multi-omic correlates. I will go to the later part later, but this was how we started to see whether the doshas can be actually linked at the molecular level. So what did we do? So we took a population, and they were all genetically related to each other, because we did not want to confound it by people saying that if you are seeing the differences are just because they come from different genetic background and nothing to do with Prakriti. So at the backdrop of Indian Genome Variation Project, we could identify a set of populations who are more related to each other. And then we identified predominant Prakriti groups and looked at biochemical profile, a genome-wide expression profile, genetic study, and then tried to synthesize the information and correlate it with how does it the molecular profile match with the Prakriti. So this is what we did. This was the genetic map, and we took populations from here, and then Bhavna designed a questionnaire which allowed us to distinguish people of predominant Prakriti groups. And then we looked at their biochemical profiles, which are used in routine diagnostics. And then we had an array of 8,000 genes in which we did the expression analysis. And after that, so we screened around 850 people with two Ayurveda doctors. There were 80% concordance in them, along with a software analysis to be able to identify 96 extreme Prakriti types. They were genetically tested that they all came from Indo-European background and not confounded by mixing from any other population. And then we looked at what we find. What do we see as differences? So we find there were differences with respect to gene expression. So this is just a representation. I will not deal with in much detail. You can look up at the original paper, but basically the green shows down regulation, the red shows up regulation, and this is up in Vat, up in Pit, up in Kapha, down in Vat, Pit, and Kapha. This is in males and this is in females. So you see, they differ with respect to response to stimulus, metabolism, regulation, development, transport. All these processes differ between the Prakriti type. More immune response is higher in Pit and lower in Vat. Building up like biosynthesis is more in Kapha. Physiological process, fibrinolysis is lesser in Cough and then cell proliferation, etc., are more in Vat. So these are the first kinds of observations we found. And there were many systemic links. So one gene governing multiple functions, multiple diseases, those also panned out to be differing between populations. So this is a summary that what we found was, if we take any biochemical parameter, normally there is a broad distribution, but we found Vat, Pitta, Kapha were in limited in their distribution, so that Kapha will be restricted here. So any movement from here to here will be a disease, but it would be normal for Pitta. And here some parameter will be normal, but higher could be a disease. So that's how we found different biochemical parameter to differ. These were like lipid profiles, hematocrit. So hematocrit was higher in Pitta, lipid profiles were higher in Kapha, and we found processes related to cell cycle, immune response also differentiating. So here you can find that Vat governing. So this was our first paper, and this is what was the inception of Ayurgenomics. And why I call it as Ayurgenomics is this one, because cell cycle differing between Vat, Pitta, and Kapha, and cell cycle vividly described in Ayurveda to be a function of Vat, we are able to get a molecular interpretation of a Sanskrit shloka. So when I talk about Ayurgenomics, it is a language. It is just a molecular language which can bring the Ayurveda text to mainstream. So Parmanu Bhed Parisankhya work of Vayu is vividly described in terms of Vat. Now, if I, so this Vat, cell division, gross phenotype, all were linked by Ayurgenomics. How does it pan in modern times? So if there is a database where you can look for abnormality of cell cycle, you find that there are certain diseases which are governed. So Fanconi anemia is because of abnormality of cell cycle. And if you look at the phenotypes of Fanconi anemia, then you see weight loss, growth delay, short stature, intrauterine growth retardation. All of this, if you ask an Ayurveda doctor, they will say that these are Vat phenotype. There are more to it, but predominantly the phenotypes which are described. So now here you see a defect in cell proliferation leading to a disease, and that disease has a again phenotype which are more like Vat Vikriti. And in today's day, people are now finding that cell proliferation is also linked to autism. People are talking about in the prenatal environment, if there is more cell proliferation, then children have a big head size and macrocephaly. And it seems to be autism is more because of what happens in the prenatal environment. And people have also shown that if you take autistic children and you make their their cell lines, you find that there is more replication, more DNA damage. And now people are also talking about relationship between cell proliferation and morphology of the face, which actually is molded in the development. 10 years later, we also did an exercise where we, from a different cohort, we took peripheral blood and took the cells of those and immortalized them to look in the Petri dishes. What is the behavior of the cells if derived from Vat, Pitta, or Kapha? These are biological replicates, three of each of them. And we find that Vat divides much faster, so the doubling time is smaller compared to Kapha. So we were curious as to why Vat, if they have so much cell proliferation, why is the body size still smaller? So what we did was we gave them a UV damage, and we find that there's more death in Vat and Pitta compared to Kapha. But if we wait for some time, then Vat numbers become similar to Kapha. So it seems to be that cell proliferation is a protective phenotype for Vat because they are more prone to DNA damaging. So they have to keep on proliferating faster to be able to not lose all the cells. So this shows that maybe cell proliferation is a trade-off in Vat, and that also is described in Ayurveda. But this has also got a relevance that supposing there was a cancer patient derived from, depending on the person, if the person is Vat, Pitta, and Kapha, and you want to do precision chemotherapy, and you take the cells from Vat and you find more death of the cells, then you might treat them with lower amount of chemotherapy, but then what could happen is there might be more recurrence because it actually has a higher proliferative capacity. So these are testable, but these are the places where Prakriti might become very important. So I'll finish here today that hopefully I've been able to convince you that there is a way by which we can actually integrate Ayurveda with modern or mainstream through this Ayurgenomics approach, which could also be a new way of threading molecular variability. So you have so much variability in the genome, which can be threaded through cellular and biochemical studies and linked to these phenotypes, which some of them are measurable. And then that could explain why people are different at the organism level, and those would be present in all the populations of the world. So I would like to close here. It all started with the vision of Professor Brahmachari, who happened to visit an Ayurveda seminar and came up with a triangle and showed me Path, Pitta, and Kapha and asked me to look it up. And then Bhavna joined, and 20 years we worked together. She's an MD in Ayurveda, and she could actually, I could converse with her, and we could build this field together, along with a large number of students who belong to Ayurveda, computational biology, and a large bunch of collaborators whose work I'll describe in the coming series presentation. And it's been funded by uh, uh, Ministry of Ayush, CSIR, and now I'm over here in IIT and I'm doing Ayurtech. So that will come to a bit later. I thought that today I should give you the beginnings of how Ayurgenomics started and what I would like to uh, mention what is Ayurgenomics. So anything you take and do use genomics would not be Ayurgenomics unless you are able to get the meaning across to Ayurveda iteratively. So that is what I would define Ayurgenomics as. Thank you so much. So I'd like to close now.
Thank you so much. This was such a wonderful presentation. Lots of information, very well presented. Thank you so much, Dr. Mitali, for such an informative, very, very valuable presentation. The entire journey of development of human genomics, the variations, the technological advancement, and most importantly, how to connect Ayurveda's core concepts of dosha, prakruti, Sutra, individual variabilities with the modern concept of human genomics. It was very well presented. Thank you again for taking the time to, for sharing your years of experience and work with every single attendees. We are very excited for the remaining series, and thank you everyone for attending our webinar. Please watch and subscribe to our YouTube channel to view earlier webinars and other presentations. Please like, comment, and share our videos. Once again, I invite you to become our member and also fill out our volunteer form. You can also subscribe to Ayu Update newsletter and stay tuned with our projects and the events that we do at GCAR, Global Council for Ayurveda Research. If you have any questions, feel free to contact us at ayurvedaresearchusa@gmail.com, or you can also reach out to me at asavari@mnayurveda.com. Thank you again. Thank you so much.
Thank you. Thank you so much. Thank you.
So our next session will be next month around the same time. It will be on March 20th, 9:30 a.m. EST. So that is India, 8 o'clock IST.
Okay.
Thank you so much.
Thank you.